Hello students, myself Popere KS, Assistant Professor, Botany of Ray Shikshan Sanstha's RBNB College, Srirampur. In this lecture, we are going to study about the gene regulation in case of eukaryote. We have already discussed the gene regulation in case of the prokaryote, that is the lac operon. Now in this lecture we are going to discuss the gene regulation in case of the eukaryote. So the gene regulation in case of the eukaryote is very different from that of the prokaryote. So this gene regulation or the control of the gene expression, it occurs by two different processes, that is first is short-term regulation or the reversible regulation. So this will be there for a short duration of a time. Next is the long-term regulation. So this regulation will be there for a longer duration. So let us first see the short-term regulation or the reversible regulation. So it will include the various processes which will involve the activities of the different enzymes or the mechanism which will be occurring and it is depending on the concentration of a substrate. So the formation of the enzyme or the synthesis of a particular protein or the different mechanism, it will be depending on the concentration of a substrate. As the substrate will increase or decrease, ultimately it will lead to the decreasing or the means the process will slow down due to unavailability of the substrate. So this is the short term regulation. Now this will involve the different processes, like in case of the cell division. During cell division, what will occur? There will be a rise, or we can say it may rise, or the quantity or the amount may decrease depending on the availability. Suppose so the concentration of the amount or the concentration of the amount of the DNA, RNA or the protein which is synthesizing, it will be varying depending on the situation or the time. So the rise and the low of this protein, this comes under short term regulation. So during the cell cycle, this will occur. That means the quantity of this RNA, DNA and protein, this will be varying, or we will see a fluctuation in the level of this DNA, RNA and protein. So this is about the short term regulation. Now next is long term regulation. So this will involve the different processes which will be occurring during the development or the growth of an organism, or also in case of the plants also. So this long term regulation, it will involve the processes which is related to growth and development, like determination, differentiation also, de-differentiation. So all the process or the activity which are related to the growth and development of an organism or a plant, or we also can say during the development of from an egg to the fully developed organism. So all the steps which will be involved in development of an egg to an from egg to a complete organism, all the steps which will be involved will comes under this long-term regulation. So now next we will study a model which is given by, that is the Britann and Davidson model. This model is also called as the gene battery model. So this is Britann and Davidson model. This is also called as the this model, it is comparable to the earlier we have studied about the lac operon in case of the prokaryote. Likewise, this gene battery model, it can be comparable to the lac operon model. In this model, there will be different types of gene which are involving, that is like structural gene, regulatory gene, depending on that this model works. So this is the structure of a gene which will be involved in the gene battery model. So it consists of 4 different genes that are these are the 4 genes which are involved in the gene battery model. So first is the sensor gene, next is the integrator gene, after that there is presence of receptor gene, and last one is producer gene. So during this model what will happen, firstly this sensor gene, it will receive any inducible signal, or the signal may be in form of any hormone, protein or an enzyme. So this sensor, it will receive any signal that may be any hormone, enzyme or any protein. So what will happen, this hormone, enzyme, it will bind to the first to the sensor gene. Now what will happen, this will become active. So So now the first sensor gene, it has become active. Now what will happen? This sensor gene, it will give signal to the integrator gene. Now what will happen? This integrator gene, it has received the signal by the sensor gene. Now this gene will get activated. This will produce a mRNA. Now this mRNA is produced. This integrator gene, it is comparable to the regulatory gene in case of the lack operon. Now what will happen? This mRNA, this integrator gene now will give signal to the receptor gene. So this mRNA, it will come and bind to this receptor region, or it is a signal to the receptor gene. Now what will happen? This receptor become activated and it is prior to the producer gene. So what will happen? Now this receptor, as it has become activated, it will give signals to the producer gene. This receptor gene, this is also comparable to the lac operon, the operator gene which is present in the lac operon. This gene is comparable to that gene, that is the operator gene in lac operon. Now next, what will happen? Now this receptor gene which has become activated, it will give signal to the producer gene, that is the last gene. Now what will happen? This is a gene which can be comparable to the structural gene of the lac operon. Now what will happen? After receiving the signal, now this producer gene, the mRNA of this producer gene will be synthesized, and after that there will be protein will be formed by this mRNA. So this is the end product that will be result of the activation of the producer gene. So these 4 genes, that is sensor gene, integrator gene, receptor gene and producer gene, they will work in a synchronized manner. So this is the Britann and Davidson model. This is also called as gene battery model. So this is about the gene regulation. In case of the eukaryote, it takes place by two types of regulation, that is the short-term regulation and next is the long-term regulation. After that, we have studied the Britann and Davidson model, gene battery model. So it involves the 4 genes and they works in a synchronizing manner. So this is about. Thank you.